Assalamualaikum to Dr. Muhammad Izani. We are from class B2 HS1113A1 and our topic for CSI 256 assignment is Enzyme-Linked Immunosorbent Assay. I would like to introduce my team members, which is Clarissa Eva, Farah Shamirahana, Fatinur Shahira, Fatinur Farisha and also Hanis Masharaf. First of all, I will start with presenting the introduction and history of ELISA. Enzyme-Linked Immunosorbent Assay, or known as ELISA, is a method to detect and quantify biological substances such as protein, peptides, antibodies, and hormone. This immunological test is very sensitive. ELISA test has been utilized as a diagnostic tool in medicine and plant pathology, as well as a quality control check in a variety of industries. ELISA can give a quick and useful measurement of the concentration of an unknown antigen or antibody by combining the specificity of antibodies with the sensitivity of a simple enzyme assay. Antibody is linked to an enzyme, and the substance is added in the final stage that the enzyme can convert to a detectable signal, most frequently a color change in a chemical substrate. ELISA test is commonly used to diagnose HIV infection, pregnancy test, and also blood typing. Now, let's move to the history of ELISA. It all started in 1941, where Albert H. Kunz and his colleague became the first to label antibodies with fluorescent dye and use it to identify antigen in tissue section. This method known as immunofluorescence. Then, in 1960, Rosalind Sussman and Solomon Barrison introduced radioimmunoassay. However, due to radioactivity posing potential health issues, researchers need to find a safer alternative. Next, ELISA test started to be introduced in 1971 by Eva Engvall and Peter Perryman. This method uses antibody to seek out the presence of hormones and viruses. In 1976, competitive ELISA was invented, which a conjugated substrate competes with a protein of interest. It was developed and used to detect human choriogonadotrophin hormones. Moreover, in 1977, there was a method which the detection antibody is coated onto the plate surface before the protein of interest is added. It is developed and tested on several substrates for proof of concept. This method is known as sandwich ELISA. In 1978, indirect ELISA was developed to detect human serum albumin by adding a secondary antibody to for detection purposes. And in 1985, ELISA test is the first screening test that commonly employed for HIV. Lastly, ELISA is used to test for antibodies of coronavirus in today's pandemic era.
Next, I will be moving to the procedure. Firstly, direct ELISA. An antigen is coated directly onto the plate's walls and incubated it for 1 hour at 37 degrees Celsius or for overnight at 4 degrees Celsius. Once incubation is complete, the plate needs to be washed to remove unbound antibody. Then, antibody conjugated with enzyme is added and bind directly to the antigen. The ELISA plate will be washed again to remove any unbound antibody. And lastly, substrate is added. Color product is developed due to the reaction of enzyme bind with the substrate. Secondly, indirect ELISA. Same as direct ELISA, it started with an antigen is coated on the plate's wall, incubated for 1 hour or overnight, and once done, the plate needs to be washed to remove any unbound antibody. After that, the primary antibody is added, followed with wash step. Then, the secondary antibody conjugated with enzyme is added and incubated once again. Before adding the substrate, the plate will be washed again. Colored product is developed due to the reaction of enzyme bind with the substrate. Next procedure is sandwich ELISA. This type of ELISA started with captured antibody coated onto the plate's wall and incubated for overnight, followed by washing step. Next, the antigen is added to bind with the captured antibody and incubated once again. After the ELISA plate is rewashed, the primary antibody conjugated with enzyme is added and the plate is rewashed again. Lastly, the substrate is added and color develops. The last procedure is competitive ELISA. This type of ELISA uses two specific antibody, which are an enzyme conjugated antibody and another antibody present in the test serum if the serum is positive. When the two antibodies combining into the wells, this allows a competition for binding to antigen. Competitive ELISA started with the captured antibody is coated onto the plate's well. Antigen antibody is added subsequently, followed by washing step to remove any unbound antibody. Then, the secondary antibody conjugated with enzyme is added and the ELISA plate is rewashed. Lastly, the substrate is added, and if there is color changes, it can be interpreted as negative result for the reason of enzyme conjugated antibody bound with the antigen, not the antibodies of the test serum.
So next, we will go to the application of ELISA. The first application is ELISA applied in disease outbreak. It is to keep track of disease outbreaks, for example HIV, bird flu, STD, and many more. ELISA has shown to be a reliable platform for detecting a wide range of disease in humans and animals all over the world. The second one is pregnancy test. ELISA can detect HCG, one of the most frequent hormones during the first month following fertilization. In the early stages of pregnancy, ELISA can determine some of these proteins in the maternal blood, saliva, or urine. Last but not least, for application of ELISA is vaccine development. An ELISA test is used to create a vaccine. Antibodies against antigens that were purposefully injected into an immunized animal or human model can be detected in the serum of the immunized animal or human model.
Limitation of ELISA. Multiple wash step. Inadequate washing can cause poor agreement among replicates because of the large variation across the wells make it difficult to fit the linear standard curve or could introduce large error bars to the sample confounding the result. Detection of weak interactions. Weak antibody protein interaction may not be detected due to multiple wash steps in ELISA assays. High background. ELISA has potential for high background which impairs the sensitivity of the assay. It could come from poor washing steps, TMB substrate contamination, or cross-reactivity. It can lead to false negative or false positive data. Need large sample volume. It will hinder how many targets can be quantified from test sample and severely limit the ability to add replicates for more reliable results. Time to results. The assay time is generally took about 4 to 6 hours, not including overnight coating of the assay plate with captured antibody. Stability of the signal requires reading within minutes of adding the stop solution, even the reaction often not fully stopped. The data will drift over time as seen with additional reads of the plate. ELISA alternative technologies. Amplified luminescent proximity homogeneous assay, alpha assay, low background, faster time to result, low sample volume, strong detection of weak interaction. Time resolved fluorescence resonance energy transfer, TR-FRET, long signal stability, low sample volume, no wash, faster time to results.
Behind the sophistication of the technology, there are definitely advantages and disadvantages. The advantages, there are numerous sensitive assay equipment offered. Second one, there are no radiation risk. Third one, reagents are inexpensive and have a long shelf life. The last one, ELISA can be applied to a wide range of biological samples such as plasma, serum, urine, and cell extract. Disadvantages, first, as matched pairings, only monoclonal antibodies can be utilized. Next, if the blocking solution is ineffective, negative control may imply positive results. The last one, because the enzyme or substrate reaction is short-term, the color must be applied as soon as possible. This is a summary for our task ELISA. To conclude, ELISA is a unique technique for detecting an antigen or antibody present in a biological sample, both qualitatively and quantitatively. Second, the approach is frequently utilized in diagnostic and drug screening, despite its drawbacks. The last one, the chromogenic detection method employed in ELISA is easy, sensitive, and risk-free for any assay. These are the references we use to find information about the task given. Lastly, from our presentation is evident to prove that we work together and work hard to complete the assignment given as best as we could. That's all from our group. Thank you.